Now let's talk the CTAB protocol DNA extraction. First, let's face the CTAB protocol materials: 1 CTAB, 2 sodium chloride, 3 10% SDS, 4 24 ml, chloroform 2, 1 ml, isoamyl alcohol, 5 isopropanol alcohol, 6 proteinase K, 7 adida, 8 cold ethanol alcohol, 70% to 100%, 9 beta-mercaptoethanol, 10 Tris-HCL and Tris-base. So, to prepare 100 ml extraction CTAB buffer with 8 pH, the components will be 1, 5 molar, sodium chloride, equal, 28 ml, 2, 1 molar, Tris-HCL, equal, 10 ml, 3, 0.5 molar, adida, equal, 4 ml, 4, 2%, CTAB, equal, 2 grams, 540 ml, distilled water, 6.2%, beta-mercaptoethanol. The component of phenol, chloroform and isoamyl alcohol equal 25 to 24 to 1, respectively, and the functions of these agents abbreviated in the following points: 1. Phenol denatures proteins, and precipitates format interface between aqueous and organic layer, while chloroform, increased density of organic layer, and isoamyl alcohol, prevents foaming. On the other hand, the lab equipments for CTAB protocol consist of 13 pieces: 1. Pestle and mortar, 2. Digital balance, 3. Micropipettes with different volumes, 4. Micropipettes tips, 5. Micropipettes tips racks, 6. 0.5 to 1.5 to 2 ml sterile Eppendorf tubes, 7. Eppendorf tubes racks, 8. Sterile glassware, 9. Water bath, 10. Vortex, 11. Centrifuge, 12. Freezer, -20 Celsius, and finally, laminar flow cabinet, as showed in the following slides. The overview protocol of CTAB DNA extraction, including 7 points: 1. Collect fresh plant material, 2. The plant material is mixed with extraction buffer and ground in a mortar and pestle. 3. The resulting liquid is centrifuged to separate the solid plant material from the extraction buffer supernatant. 4. The supernatant is mixed with ethanol to precipitate the DNA, which will form a pellet at the bottom of the 1.5 ml Eppendorf tubes. 5. The supernatant is discarded. 6. The DNA pellet is washed with a 70% ethanol solution. 7. The DNA pellet is dried and then dissolved in tea buffer or distilled water. In the same line, in details, the CTAB protocol of DNA extraction start with breaking down the cell wall and the membranes of the plant materials. Then, make centrifugation, to separate the solids from the dissolved DNA, and the precipitate of the DNA will be done by using isopropanol alcohol. After that, use centrifuge to separate the DNA from the dissolved salts and sugars, and finally, wash the DNA pellet with 70% ethanol, then, dry pellet, and dissolve the extracted DNA with TE buffer, or distilled water. As viewed in the next slide. Now, let's face the tips in resuspending DNA pellet. Firstly, use pipette tip to break up the DNA pellet to ease resuspension, and do not make pipette up and down repeatedly during resuspension. This break DNA. Then, leave DNA on refrigerator overnight to let it slowly redissolve. And now, the DNA solution ready to use, but be attention please, to keep DNA solution stable, keep it in the fridge for short-term storage, and in the freezer for long-term storage. After that, run the extracted DNA on agarose gel to check its integrity and quality. By following the next steps: 1. Mix 10 microliters of DNA with 10 microliters of loading buffer. 2. Load this mixture into a 1% agarose gel. To calculate the amount of DNA or RNA in any sample, must be follow the next equations. For DNA, the equation equal, OD, 260 by 50 nanogram per microliter by dilution factor, while, for RNA, the equation equal, OD, 260 by 40 nanogram per microliter by dilution factor. So, the pure preparations of DNA and RNA have OD, 260 divided by OD, 280 values of 1.8 to 2, respectively. So, the bases in DNA divided by RNA absorb UV light at max at 260 nanometer, as showed in the following equations. 1 OD at 260 nanometer for double-strand DNA equal 50 nangram per microliter. 1 OD at 260 nanometer for single-strand DNA equal 20 to 33 nangram per microliter. 1 OD at 260 nanometer for RNA equal 40 nanogram per microliter, as showed in this chart. Therefore, the DNA measurements procedures that done by spectrophotometer are: dilute the sample of purified DNA by add 700 microliter of T buffer to the purified DNA. Then, measure the absorbance at 260 nanometer. After that, measure the absorbance at 280 nanometer, and assess the DNA purity by divided 260 to 280. The accepted ratio of this divided is between 1.7 to 1.9. For example, calculate the DNA concentration. The provided A260 equal 1, and the provided A280 equal 0.5, the DNA concentration equal 50 nanogram per milliliter. So, the unknown DNA concentration can be calculated by cross the multiplication to get the final DNA yield. From this equation, the final DNA yield equal DNA volume by DNA concentration. Another example: if you have volume of DNA solution 100 microliter, and the DNA concentration is 30 nanograms per milliliter, the DNA yield in nanogram equal the volume by the DNA concentration. This slide showed the extracted genomic DNA on the agarose gel. The extracted genomic DNA has very high base pair, and the band at the top of the gel, near the well, mean the DNA was extracted correctly. So, be attention please, poor quality of DNA will not perform well in the PCR procedures. This slide showed the different shapes of DNA on the agarose gel. The first one showed there are some DNA left in the well, the second shape showed the sharp band, while the third shape showed no sign of proteins. On the other hand, the fourth shape showed there are no smear of degraded DNA, and finally the fifth shape showed no sign of RNA. All of these shapes can be known by using nanodrop device, which give us the results of each sample according to the ratio of 260 to 280, which equal 1.8 to 2, or the ratio of 260 to 230, which equal 2 to 2.1.